set the stage for our discussion, you could maybe provide just a quick overview of the pipeline as it stands today, and maybe just a few of the upcoming catalysts that investors can expect in 21.
Yeah, happy to do that. A decade after the CRISPR-Cas9 platform was elucidated and developed by Dr. Emmanuel Charpentier and Dr. Jennifer Doudna, we're very pleased to be at the forefront of leveraging this exciting platform for groundbreaking therapies. In a very short amount of time as a company, in about 6 years, we've gone from starting with a research platform, to having multiple candidates in the clinic. Our lead asset, CTX001, in hemoglobinopathies have shown remarkable data and could be potentially curative for patients suffering from sickle cell disease and beta thalassemia. And we'll talk more about this program, but we continue to make progress in that program to get that program to patients in a widely accessible manner by getting to filing and approval. With our oncology programs, we have 3 different CAR Ts. These are allogeneic CAR Ts. That means that they're made from healthy donor T cells, and designed using the CRISPR platform to target the cancers and kill the cancers in patients suffering from diseases like lymphoma and even solid tumors like renal cell carcinoma. Those programs are all in the clinic, and we will have additional data for CTX110 this year, as well as initial data for CTX120 and CTX130. 110 being targeted towards CD19, 120 being targeted towards BCMA, and 130 being targeted towards CD70. Beyond that, we have a very rich pipeline, starting with our diabetes program, where we create artificial pancreas from iPS cells using CRISPR, and that should go to the clinic out of this year, and a number of in vivo programs that are also moving towards the clinic rapidly. So very full pipeline. We're very excited with everything that's going on. We're over 450 people now as a company, and expect to keep marching on, especially as our own manufacturing facility comes online next year.
Great. Thanks for that. Obviously with EHA coming up next week and the data update in sickle cell and beta thal, maybe provide just a little bit of context on what to expect in terms of data updates. Obviously, you provided updates in enrollment recently in the trial. And then maybe, after the ASH data, if you could provide any color on how physicians are looking at the data set to date, and how enrollment has proceeded since that data update at ASH last year.
Yeah, I think ASH last year was a very important milestone for the program. I think it was truly seen as a proof of concept or POC point for thalassemia and sickle cell. We presented data there for 10 patients, 7 thalassemia and 3 sickle cell patients, and all 10 patients were symptom-free. In the case of thalassemia, those patients didn't require a transfusion anymore within a couple of months of being treated with CTX001, and all the sickle patients were free of hospitalizations or vaso-occlusive crises. And that's a remarkable difference for these patients that were been living with serious disease their entire lives. So that was seen as a big, sort of proof of concept point. And subsequently, what we've disclosed is that we've dosed more patients in our EHA abstract. There's a greater number of patients. We have 14 patients, 10 thalassemia, 4 sickle cell patients. And we also said that we dosed 30 patients now. So I think that the program is getting more and more momentum. Both trials are designed to enroll up to 45 patients. But we don't even think that we may need 45 patients worth of data if for approval the data continue to bear out and hold out. So I think we look forward to providing continued updates as we go along at this conference and next.
Great. And then there's obviously a lot of development in the ex vivo HSC space with sickle cell. Maybe just provide a little bit of color on how you're viewing CTX001 in the context of the clinical development landscape. Obviously, a bunch of different modalities looking at HBA87Q, HBF, and HBG Macasa, for example. So just be interesting to think about as the leader in your particular modality, how you're thinking about your program as well as potential competitor programs that might get advanced in the clinical trials in the future.
Yeah, there are a number of programs in the space, but I think the advantage we have at this point that positions us as a clear leader in the space is the fact that the data with CTX001 are quite remarkable. I think every patient so far has been free of symptoms. As you compare that to lentiviral approaches, the data look better. And when you combine that with the fact that CRISPR is a notionally safer approach relative to random integration with lentivirus, I think you'll have a huge advantage in the marketplace. So I think you have, 2 players now neck and neck at the front. But we believe that CTX001, especially with the capabilities that Vertex brings to the table in terms of commercialization globally, we would have a significant advantage. I think there are a number of players that are following in our footsteps. But I think, in this market, time is an important component. I think it's going to be difficult for players to get a commercial foothold if there's been a player existing already for 4 or 5 years with very good data, right? I think this is like the medical device space. I think shares are sticky because there's certain centers learn and train themselves on a certain product, and it's not very easy to switch. So I think while it's, it won't be difficult for people to enroll patients because there's a lot of demand for gene editing in sickle cell and thalassemia, I think commercially we are well positioned to take on that leadership mantle and maintain it over time.
Great. And you mentioned Vertex, and I wanted to kind of drill into that a little more. I think it's a very innovative partnership, and obviously the recent deal that you guys did to amend the partnership provided the company with significant upfront capital for fairly minimal economic terms. So maybe can you just talk about how the Vertex collaboration started, and then with the most recent deal with the negotiations over the 10% and the control of the launch, how you were thinking about it from CRISPR's perspective?
Yeah, it all started with ASH conference last year. We presented data and we said, oh my God, look at the response from physicians, from the patient community, the patients themselves. While we had exciting data with 1 patient in thalassemia, or 2 patients in thalassemia, 1 in sickle earlier, having 10 patients worth of data made a big difference. I think, everyone was looking at the program very differently and saying, gosh, this can be a huge advance in the field of medicine. And I do think that 20 years from now, we're going to look back and say that was the moment, ASH 2020, when these data were presented that marked the arrival of a new class of medicines that may change the face of, change both delivery of care with curative therapies and change the way we think about diseases and amelioration. So I think as we looked at it, both Vertex and us said, how do we make sure we execute very quickly and get to a global launch? And in the original construct, we had CRISPR as the commercializing party for the US with Vertex for ex-US. But in this context, as you look at the data, the trials are global. We're looking at the market in a way that, there's a lot of shared responsibilities from a central global marketing, I would say, versus regional. It made sense to have a coordinated launch, to have one company lead it, whether it's CRISPR or Vertex. And I think, just looking at it from a capability standpoint, it made sense to align the capabilities in a way where you can leverage all of what Vertex have to get this globally coordinated launch and bring their best practices to bear from cystic fibrosis and other markets to ensure that we can reach as many patients as we can. In the meantime, I think, CRISPR's capabilities are best harnessed thinking about, what's sort of the next horizon, which is how do we come up with gentler conditioning agents, further expand the market? How do we come up with improvements in process and analytical capabilities to that so that we can, further streamline that patient experience and improve outcomes for patients. So I think it aligns capabilities well. The capital does make a difference for us as we further expand the rest of our pipeline. And most of all, I think it makes sense for patients suffering from sickle cell disease and thalassemia, because this is the best chance of making sure that this program gets wide access to all these patients that need and deserve the therapy.
Yeah, and obviously you touched on this a little bit, but the global manufacturing cohesion that you have, can you maybe just describe a little bit about how to view that in the context of the rest of the pivotal or the rest of the current clinical trial, as well as approaching the agency, just given some things that we've seen in the space recently with regards to regulatory filings with the lentivirus procedure. It'd be interesting to kind of hear how you're going to be approaching it, learning from some of the aspects of the first mover has had to go through.
Yeah, I think one of the advantages we have is right from the get-go, we said we're going to do one global trial. We're not going to separate a trial for US and Europe. We also said from the get-go, we're going to have one manufacturing process that can be scaled into commercial versus starting with something that's more of an experiment, right? So I think that, combined with the fact that the regulators are quite closely in touch across the US and Europe, it gives us that ability to sort of advance this as one global trial, ultimately with the notion that the filing package would be similar or same across the US and Europe. Now, I think the rest of the world and Asia is a different story. But at least at this point, that plan seems to hold true. We have the RMAT designation in the US, which gives us the ability to have frequent dialogue with regulators in the US. and PRIME designation in Europe, which also gives the same ability to speak with the EMA folks quite regularly. And I would say that everyone's been very supportive. Now, ultimately, when it comes to commercial and reimbursement, I think that'll be different by region, right? I think you'll have a different way of looking at reimbursement for maybe, Germany versus Italy versus the US. And there we have to have a more nuanced and bespoke strategy. But otherwise, I think this notion of global trials makes a lot of sense. It allows us to move faster and allows us to make sure that we have a very coordinated package.
Yeah, no, that's helpful. If we could switch gears a little bit into the oncology pipeline, obviously, going to be 3 separate readouts for 2021 from this pipeline. With regards to CTX110, you presented some initial data there, seeing a 50% CR rate, pretty impressive proof of concept in T cells now, building on the HSC platform. What should we expect in terms of updates on the 110 program? Obviously, going to be a focus on more on durability now from those initial patients, but maybe set the stage on what to expect from CTX110 in the update, and then maybe we can touch a little bit on context with some of the other players in the space.
Yeah, I think data from the different allogeneic players is good. It just basically signals that allogeneic therapies are here to stay. You're seeing response rates and CR rates that are similar to autologous therapies, right? I think that's something that's important. And in fact, you're seeing some notion of durability. The trials are still early where you can't really compare apples to apples, but there is a notion that patients can be in CR for a long time based on allogeneic therapies. And the big switch, I think people are starting to understand allogeneic therapy as different from autologous therapy. I think we used to get a lot of questions of CRS and ICANS. I think that's, now people understand that may be more of an autologous phenomenon than allogeneic, because you just don't have the same kind of expansion. I think you saw something yesterday in the news around ICANS with autologous therapies, but you may not necessarily see that with allogeneic therapies, even if you have the same potency of killing, right? I think you have cell expansion relate as the allo CAR Ts go in there and kill the cancer cells, but it may not be the same profile as an autologous therapy, right? And in fact, even as you look at autologous therapies, there's the correlation. There's some correlation expansion to CR, but it's not a perfect correlation. And so we're learning more about CAR Ts in general, and we're also learning more about allogeneic versus autologous therapies, and the same sort of metrics and lenses that you apply to autologous may not apply to allogeneic. And what we've learned is that, a year ago, there were probably 2 schools of thought. There's like this notion that you're going to redose every time you progress, and that's how you can ensure durability. And now there's this notion that you can redose, preemptively as a consolidation dose, and that's the way to get durability, because you've completely eliminated the cancer. And I think I'm glad the field is pursuing both those options to see what's better. But in all eventuality, you're going to get allo therapies approved in the late line settings, and once you're getting to approval, it's going to leapfrog autologous therapies, because it's going to move much faster. It's easier to enroll patients in allogeneic therapies versus autologous therapies. Imagine trying to expand your autologous cell therapy trials to community settings. It's just very slow. You're not going to get there. Now, all that said, I think, as we look at the data for ourselves versus other companies, there are many different ways to look at our data. Eventually when we disclose the data, you'll have the basis for comparison. But I think the beta 2M strategy gives us that persistence that's important for killing the cancer cells. And I think ultimately maybe a safer approach with flu-cy as conditioning relative to adding CD52 antibody. I think those are all things that will be important to compare and contrast as we go along. But it's less about us versus another company in allogeneic. It's more about allogeneic versus other modalities. And I do think the Sheen's worn off some of the bispecifics. If you think about ASH last year or where we were last year at this point, there was a lot of excitement about bispecifics. Bispecifics have the same toxicity issues that autologous therapies have. And I do think allogeneic therapies are well poised now to become a very important part of the treatment landscape in lymphoma and other diseases.
Yeah. One quick question on 110. I mean, you mentioned the consolidation regimen. Is that something that you might look at with 110, or is that something that you would need to add a increasing grafting window to do something like that?
No, I think our hope is still that a one-time dose gives you a durable response. Right? I think that would be ideal if you can do that, because then there's no argument really for doing autologous therapies. But I think if we do need to go there, we will probe the consolidation dose regimen if we do need to do that. And we're obviously watching carefully for data from other players to see what the consolidation doses do, and what are the relative kinetics of it, what are the relative dynamics of it in terms of tumor volume reduction and amount of doses you need, et cetera. So we're learning a lot, and we'll go there if we need to. But at this point, we want to structure an experiment to see what does a one-time dose do in terms of durability, and is that competitive versus autologous, and is that competitive enough to get single arm approval if you can expand to a pivotal trial.
Right. Yeah, I mean, I think the initial data clearly de-risked the B2M knockout approach, which was the big question going into your first data set. So this one will be interesting to see, the durability and the persistence of the cells, and answer those questions. With 120, the BCMA targeted product, I mean, obviously, response rates are numerically higher with the autologous programs. Is that something that you think is a differentiator with 120 versus 110? I mean, how are you looking at your 120 update in the context of autologous, and maybe compare and contrast that with CD19, which is a little different?
Yeah. And I think the 120 CR rates seem to be higher, right? I think, or BCMA in general, and especially with the data that came out from Legend yesterday. I think you're looking at a 2 and 3 type CR rate with autologous BCMA programs, which is better than some of the other programs out there today. So that does set a bar that's relatively high. The durability in BCMA has not been that great compared to CD19 in some cases for the trials where we know the data. I think it'll be interesting to see what the median duration of response is for the CRs for the Legend J&J program. That would set the bar. Right. So I think, do you have to exactly meet the bar to be viable as an allogeneic therapy? Unclear. You have some wiggle room, you have some latitude. So let's say, you have a program with 55% CR rate relative to 65% CR rate, there's 10% delta. These are all small numbers. That, may be sufficient to get a foothold in the market, because, you have a more convenient approach with allogeneic therapy. There are a lot of patients that don't have access to autologous therapies. And I think, the allogeneic therapies can always improve it over time. So I think there's that notion. I think, but that said, I think you do need to be close to the bar. You can't be too far off the bar. Otherwise, you're going to create an ethical dilemma for people doing the clinical trials in terms of using the available autologous therapy versus going on trials with new agents. Yeah,
absolutely. And then, obviously a few of the other companies are looking at GSI inhibitors, gamma secretase inhibitors. Is that an approach that you might think about looking at in the future, or is that something that you're just going to wait and see, how well B2M knockout works before, making decisions on anything like that?
Yeah, I think it's certainly squarely within our sights. We haven't activated that because, we want to see how it plays. Again, we want to do a controlled experiment. If we need to, we'll go that route of the GSIs. But at this point, we're trying to work with the original hypothesis, which is that beta-2M gives us that persistence. It won't be overwhelmed by the soluble BCMA, and that we still see responses. And I think it may, we'll see if we need to get Tinker and Taylor. But again, I think we need to be, have a sense of activity that's close to what the benchmark is for us to Tinker and Taylor, right? I do think that eventually there's other constructs coming out where you're turbocharging with cytokines or making other edits. Eventually, I think allogeneic therapies will get better than autologous therapies, even with the high bar that's set. But I think in the interim, you need to be competitive and get a foothold.
Yeah, absolutely. On CTX130, obviously, just the solid tumor program in your oncology pipeline. A question we get a lot is, what's the bar for success here? How are you viewing it in terms of being generating one of the first data sets in solid tumors with allogeneic, and how are you viewing it internally?
Yeah, I think this would be huge. I think we all know that 80% of the cancer market is solid tumors, and the unmet need is tremendous in solid tumors. So any hint of a response from allogeneic CAR Ts in solid tumors is a pretty meaningful step for mankind, I would say. I think it's an area that, people have been trying to get responses for a long time. As you saw with some of the news recently, there's, stalwarts in the field who've been trying to use auto CAR Ts to get any sort of response, whether it's mesothelin or other targets, and it's been difficult. And I think for us with allo therapies, especially with the CD70 target, we think is a very attractive target given the expression in renal cell carcinoma in a highly immunologic cancer. I think gives us that opportunity. I think CD70 program, the CTX130 program for us could be, a very important program in the long run. And so far we're continuing the trial. There's a lot of interest in the program. We're dose escalating as we go along, and we look forward to providing updates.
Yeah, and as far as how the medium to expect the data from those programs, you've had some where you've done obviously in concert with a medical meeting, some you've done with a webcast event, press release. I mean, how are you thinking about the medium of disclosure?
Yeah, I think generally our philosophy has been that the initial disclosure on programs has been as a company press release or a company release or presentation. I think subsequent data releases have been as part of medical conferences. We did that with 001. So I think, for 120 and 130, they're still early. I think these are all things that we determine as we go along and see what the most optimal venue is and the most optimal timing is. But we'll provide guidance as we go along throughout the year as to when to expect the data.
Yeah. This is a question on the oncology platform and the company commercialization strategy as a whole. But, obviously the CTX001 program, you partnered with Vertex and, updated that program. How are you thinking about the oncology program? Right now you have 3 wholly owned assets. Is CRISPR becoming more of a CAR T company then, if you're going to look to build out kind of the commercial infrastructure to support, global launches for that? Or do you partner one or more of those programs? How are you thinking about it now? I mean, obviously a little early, but an important question on the direction of the company.
Yeah, and part of it depends on the data, but ultimately I think our goal is to be half oncology, half rare diseases at this point, given our programs. I think we have hemoglobinopathies, and we have type 1 diabetes. We have a number of other indications like hemophilia and GSD1A, which we're working on as well. Not to mention DMD, DM1 together with Vertex. So I think it's sort of 50-50 overall in terms of oncology versus non-oncology. If the data look good from 110, 120, 130, then we have a Regeneron type platform that we can really expand and, will tilt us more in the direction of oncology. I think, but we always want to be looking at other diseases as well. We don't want to become a purely oncology player because, I think, why is that? I think ultimately it's because that's how we can exploit the platform to the fullest extent to bring transformative medicines for patients. It allows us to play across different business models. So the business models are different for other diseases versus oncology. But I think as one single entity, we do get synergy by playing across both. So I think, if you project 2-3 years down the line, I think we'll be talking about oncology data, but also talking quite a bit about, potentially about diabetes, talking about, some of the rare diseases like hemophilia, et cetera. With oncology, how we play in the long run is a question that we evaluate every so often. I think that our general goal is to take it all the way and commercialize ourselves. But that's it. I think pharma interest comes in waves. There's, I suspect, as we show data and others show data, that pharma is starting to get more interested. For a while, I think people started moving towards the bispecifics, but now I think everyone's coming back around to say maybe allogeneic is the way to go, especially as iPS cells companies abound and iPS technologies are maturing. And so, if there's a lot of interest from pharma, and it may make sense for us to do something different, right, as a structure. But that's always things that we keep in our back pocket. I think, we have a very healthy cash balance, but we don't want to be over reliant on equity financing. And we're very sensitive about dilution in general, especially because there's so many other alternative mechanisms of financing that have different costs of capital, but maybe more advantageous in the long run.
Great. I know we're running short on time, but I did want to just touch briefly on the ViaCyte collaboration, because I think type 1 diabetes is becoming more of a focus for the company going into next year, as given the TAM is so big. Can you maybe just give a quick overview of that program, how you're viewing it? And, obviously ViaCyte has the autologous version data coming out this year. I mean, is there any read through, or how are you going to look at that data in the context of the program for the allogeneic version you have with them?
Yeah, I think, ViaCyte have an allogeneic version, with embryonic stem cells, except it's not edited, right? The original version is not edited. They rely on immunosuppressive agents given together with the medicine to make sure that the artificial pancreas that are inserted into the patient are not eliminated by the immune system, right? So they're constantly immunosuppressing the patient, which is important as a proof of concept, because under that immunosuppression, are we seeing these cells sense glucose and produce insulin? That's sort of the big question mark from a biological standpoint. And if you can see that these cells indeed are capable of sensing glucose and producing insulin, that takes meaningful risk off the table, because then that would mean that, the allogeneic the version that we're working on, which is edited, is also likely to see the same efficacy in terms of production of insulin, but we have the added advantage that it can be stealth and the patients don't need to be immunosuppressed, right? At the end of the day, I think, we're delving into an area where, instead of fixing things mutation by mutation, we're just going to create a new organ and put it into the body. I think our pancreas are a banana-shaped organ but only a small portion of that is actually catering towards glucose homeostasis. And what we're doing is creating a small device that serves the artificial pancreas and inserting it into the body. You can imagine we do that with the liver cells, and these liver cells are a small device that are inserted into your flanks or in your stomach, which can serve to be a biofactory for the body. They can produce any drugs you want. They can produce any factors or proteins you want. So I think there's tremendous potential in regenerative medicine, which I don't think everybody's catching on to at this point. We essentially get very little value, I think, ascribed to us right now in regen med. But if you look at all the venture capital activity, all the places where money's going into, it's regenerative medicine, for a good reason.
Well, Sam, thank you for the time. And again, I think, the company has done an amazing job with bringing a very relatively recent scientific breakthrough into, real clinical results. And I think that's kudos to you and the team at CRISPR.